Dementia Researcher with a blog and a rating.
Hello, my name is Ajantha Abey. I am a postdoctoral researcher at the University of Oxford, and today I'm going to be reading my blog on research life hacks, things I wish I had started at the beginning of my PhD. This will be part one of a two-part blog series. There are about 1,001 articles and blogs out there about all the things people wish they had known about doing a PhD before they started. These are filled with advice like pacing yourself, embracing failure, dealing with perfectionism, managing the ups and downs, and so on. These are good and useful, but they've been written plenty of times, so this is not one of those articles. My goal here is to list several practical lab hack ideas and productivity pointers that are mostly systems that you can and should implement from the very start of your PhD, or whatever major research project you are embarking on, e.g. a postdoc or a master's research project, that will make your life easier along the way. A lot of these are data management and record-keeping tips, since a PhD is likely to be by far the longest project you'll have embarked on, and you'll need to have a good system that takes you through the whole thing and is useful to you at the end when you can't easily remember what you did 2 to 3 years ago. There are also some mindsets that I think are worth implementing as early as possible, which will constitute part 2 of this blog. Some of these things I wish I'd done from the start, some of these things I managed to do and remain incredibly grateful to my past self for. Let me know what you think. I include some cell culture specific advice at the end since this was the bulk of my actual work, but the rest of the advice is highly generalizable, I think. These systems are worth spending the time putting in place from the beginning. They will save you time and stress as you go, and by the end, they'll be habitual. The first one is to have a consistent experiment numbering system that you also use for your file numbering system. This is extremely boring advice, but it is by far the best decision I made early in my PhD, and the most frequently envied. It does not matter really what the system is, as long as it is simple, chronological, and used consistently. For my purposes, this meant that every experiment I did, I numbered and labeled all associated files starting with that number, going chronologically starting with 001. What you define as a discrete experiment is mostly up to you. For me, it was typically a coherent idea that I was testing from a given differentiation of cells, or a single thing that I was making. For example, using a single differentiation of neurons to do some calcium assays, neurite outgrowth, and lysosome assays would have been 3 different experiments. Or expanding a cell line to generate new stocks would have constituted 1 experiment. Many people start numbering their files with a year, month, date format. This can be annoying for 2 reasons. Firstly, it doesn't differentiate your files from everyone else's in a big communal database, like all the files on a common piece of lab equipment, for example, and it doesn't let you separate experiments that you might be doing at the same time. You might put your initials at the start of your file numbering to make sure that your files are distinct from anyone else sharing equipment, and you can and should incorporate a date into the file name as well. But starting with a chronologically applied number, or a number following consistent initials, will make finding and sorting everything vastly easier. Every experiment gets a unique identifier, and this identifier follows the experiment everywhere. On your samples, your culture plates, your lab book, your raw data folder, your analysis files, your images, your spreadsheets, and so on. The benefit of this is that everything stays findable and searchable. If you're looking at a graph 2 years later and you want to know where the raw data came from, the experiment number should lead you back to the lab book entry, the protocol, the sample details, the analysis, and any notes you made at the time. If the experiment numbers are chronological, they also become a rough timeline of your PhD. It also means that it's easy to put all the files related to an experiment in the same place. This sounds easy until you realize how easily things drift. You run a quick pilot experiment and then don't give it a number because it's not that important, but then of course it becomes important. Slowly, chaos can enter into the system. Take the time to maintain the system. Don't let the chaos creep in, number things. And of course, create an index somewhere obvious and easy to find so that someone later, or yourself, can come in and see exactly what each experiment number means. The second on the list is to use consistent file names and terminology. This sounds like the same advice from the previous one, but it is subtly different. A good experiment numbering system should be paired with a consistent file naming system. A useful file name should tell you at a minimum what the file is, when it was made, and what experiment or project it belongs to. This may seem excessive, but there are a few things more satisfying than opening a folder and having all of the files automatically appear in chronological order with their contents legible. By contrast, there are few things more frustrating than finding 12 files called some variation of new analysis final, analysis final use this, analysis final use this too, and analysis final actually use this one real. Consider also the following example. When doing a Western blot and stripping the membrane to reblot the same or a different portion on the blot, have you done blot 2, round 2, reblot 1, stain 2? And if you're on a gel from the same sample, is that blot 2 as well? It doesn't matter what term you use, but you should define and stick to a specific language from the beginning. In each of my Western blot folders, ideally what you'll find is the protein quantification file, for example, 002, experiment name, and BCA quantification, which I also know contains the gel maps, and then the folders for all the images, with subfolders for each experiment and then each round for stripped and reblotted membranes, and a new folder for the same sample run on a different gel. Then a file for the quantification output, and then files for the graphs, and a PowerPoint file to summarize everything, all with their own consistent terminology in their naming. And in every other experiment that involves Western blots, you'll find the same kinds of files labeled in the same way, with the same type and stage of data in each. Ideally. I'm not perfect, but it's pretty good. This is mostly about readability as it is about tidiness. It is about being able to go from a final figure back to the raw data without needing to rely on memory or vibes or lots of guessing and checking. The third thing then is to keep all your notes in one place. At the start of a PhD, it is easy to scatter notes and ideas everywhere. Some in a physical notebook, some in Word documents, some in PDF margins, some in emails to yourself, some in Google Docs, some in the Notes app on your phone, and some only in your brain. This is a mistake. Your brain is not a good long-term storage environment. Have one central place where your research notes live. This might be OneNote, Notion, Obsidian, Evernote, a carefully organized folder of Word documents, whatever you happen to use. The specific tool matters less than the actual principle. Your notes should be searchable, organized, backed up, and in the one place. It is really annoying to have to search across multiple different apps and formats of different files to find the thing you were looking for. I used OneNote extensively and found it especially useful because it allowed me to organize notes into sections, paste in images, annotate freely, and search text across different notebooks. Or across the entire notebook, really. One particularly useful feature is optical character recognition, or OCR, which means that text in images can become searchable. There's a particular advantage to things like OneNote over Notion, for example. This is surprisingly helpful because so much research documentation happens by hand. Whiteboards, handwritten notes, microscope settings, protocol scribbles, poster slides, reagent labels, plate maps, and so on. All of my handwritten lab notes, plates, important reagent labels, and so on get photographed and imported into OneNote, and then become searchable with all the rest of my other notes. The Apple iPhotos OCR is also, incidentally, extremely good, even with my questionable handwriting. This note system is by no means neat and tidy, but it is organized and searchable so that years later when you need to find something, you can. And if you follow step 2, you know what it should be called. The fourth thing then is to use a reference manager properly from day one. Your life will be made 1,000 times easier if you use a reference manager consistently from day one with a good tagging system. Zotero, Mendeley, EndNote, Paperfile, whatever you like, choose your fighter. I personally like Zotero because it's open source, free, and has a tagging system that I find extremely useful. But the important thing is not which reference manager you use. The important thing is that you use one, use it consistently, and use it well. A reference manager is not just for inserting citations into papers and generating bibliographies. This alone is enough reason to use one. It also acts as a memory system. Over the course of a PhD, you may read hundreds or thousands of papers. You won't remember them all. You remember that there was that paper about microglia and complement, or that study with the weird tau result, or that one review with the useful diagram. And using tags, folders, and notes make these papers findable again. My recommendation is to tag papers by topic, method, model, disease, and possible future use. For example, diseases like Alzheimer's, Parkinson's, FTD, topic like autophagy, mitochondria, lysosome, mitochondrial stress, heterogeneity, and so on. Models might be human brain tissue, iPSCs, human patients, cortical neurons, rodent models, primary cells, whatever. Methods used might be PFFs, IHC, single-cell RNA sequencing, EEG, PET scans, whatever you happen to be looking at or interested in. You can have lists of key genes mentioned, SNCA, MAPT, BIN1, TOM20. You can have where you might use it in your thesis, methodology, introduction, background, discussion, chapter 2, title paper, prior paper, or review paper, and other such notes, like it supports your hypothesis or contradicts your hypothesis. The most useful thing of the tagging system is that when you use search, you can combine them. Now you can search through all of your saved papers and find the ones that do single-cell RNA sequencing in iPSC models, or look at all the review papers on PFFs related to Parkinson's disease. All the papers that look at autophagy and human brain tissue in Alzheimer's disease and mention BIN1. Having tags for things that will be useful when you're writing the introduction, methods, discussion, etc. will also be super useful. A future thesis writing version of yourself will be immensely grateful if you can open your reference manager and immediately pull up all the papers relevant to a chapter, argument, technique, or caveat. It's also useful if you're planning an experiment and looking for other papers who have used the same technique. The fifth item then is to make figures as you go. Everyone says this, everyone is right, and almost always everyone ignores it anyway. Admittedly, myself included. Make figures as you go. It is not necessarily perfect publication-ready figures, but clean, labeled, interpretable figures that summarize what you did and what you found. Have them for lab meetings and conferences such as useful, but actually what is even more useful is the underlying clean data file which combines all of your different replicates and so on, and has some stats properly done on it that you can come back to later and make your final versions easily from. Additionally, on this point, if not graphs, having representative images from all your files and all your experiments makes life a lot easier down the track. You need to go find images for your final figures. Figure making takes a shocking amount of time. The graph itself takes plenty of time to format, let alone the surrounding work. Cleaning data, choosing representative images, arranging panels, writing legends, checking labels, making sure colors and fonts are consistent, finding the right statistical annotations, and then trying to figure out how to export the entire thing at 300 dpi. Making figures as you go also helps you think. A figure is not just a visual output, it is a distillation of all the work you've done so far and pushes you towards a key decision-making point. Even if the figure changes later, you will have a visual record of the project developing over time, and the underlying data will be tidy and ready for use. When it comes to writing reports or papers or talks or thesis, this is priceless. Finally then, for this part of the blog, item number 6 is having a centralized spreadsheet for everything. This brings me a bit more to the cell culture specific section, but honestly the principle applies much more widely. Spreadsheets are your friend. You don't have to make an overly elaborate color-coded monument to procrastination like I did, but having a centralized document to coordinate what you have, where it is, what condition it is in, where it has been, what it has done, what has been done to it, and what can still be done to it, and when you want to do things makes life a lot easier. My spreadsheet includes a freezer stock spreadsheet with a line for every vial of cells I've ever frozen down, and further information on when I thawed them and how they turned out, which also means you can keep a running tally of how many vials of cells you've made, for fun. I've got a log of cell passaging, i.e. notes on how many cells I had every single time I have passaged and counted cells, which makes it easy to predict in the future how fast certain lines grow, how many cells you expect to get in yield after X days in culture. I have a huge calendar spreadsheet with days of the year going left to right along the top, and cell differentiations on each row. I can mark important dates on the calendar at the top, like conferences, friends visiting, trips away, and major events, and then time experiments to ideally avoid these, and not overlap too much with each other as well. Especially when working with differentiation protocols that are months long and predictable, knowing when your big treatment or harvesting or plating days are going to be weeks in advance can help you plan the rest of your life or vice versa. I also have a log of all my protein and RNA samples and cell lysates. I did not do my PhD, but I now do religiously. This makes it vastly easier to remember all the samples you have available, how much there is left of each, how well each sample turned out, and if you need some spare sample in an experiment, what sample is going to be best to use. This requires a lot of upkeep, but is extremely helpful. I also have a page of all my plate maps for every plate of cells that I grow across all my experiments, which helps a lot in planning experiments and remembering later when sorting through microscopy files. There's also a list of every experiment, numerical and chronological of course, and what plates, lysates, samples, and so on are associated with each, and what stage each one is at, kind of like a massive project Gantt chart, with a brief note about what the main outcomes of each experiment was. There are many more possibilities, e.g. pages for calculating how to make up different kinds of media, lists of antibodies and other reagents, and their catalog numbers for easy reordering, but those are the main ones. So at this point, this blog is ballooning out, so I'll take a pause after these practical notes and curtail the useful mindsets and practices into a part 2. Most of this advice is not super exciting, unless, like me, you really like spreadsheets. It does take a bunch of time and effort and won't save you from failed experiments. However, good systems make life easier in the long run, and PhDs are a marathon. They protect you from your own forgetfulness, they make your work more predictable, more reproducible, your writing easier, your planning more realistic, and your future self less lost and confused. Set up systems, use a reference manager, and actually curate your collection. Make spreadsheets, write templates, track your samples, back up your files. Taking the time is worth it. The work now, when time is cheap, will save you effort when time is short and you're scrambling to finish. The person you will be 3 or 4 years from now, trying to write a thesis, trying to finish a paper, trying to respond to reviewers, apply for jobs, and work out what on earth happened in experiment 17, will thank you. Make current you the sort of person that future you will be grateful for. And good luck.
Thank you for listening. Join the Dementia Research bloggers and share your own views.